Assalamu alaikum. I am Usman Zafar Pracha. Here's a little introduction to CRISPR-Cas9 for beginners. Before going ahead, I would like to say that I have a presentation on this. I have an illustration on this topic, and the link to that illustration is presented in the description. Opening of that illustration along with this presentation could help you in further understanding the topic. Nevertheless, CRISPR-Cas9, here CRISPR is the abbreviation of clustered regularly interspaced palindromic repeats. The palindrome refers to a word that reads the same backward and forward, as for example radar. Forward R-A-D-A-R and backward R-A-D-A-R, again radar. In case of strands, there are also certain palindromes, as for example there is a strand 5-GAATTC3-. Then there is a palindrome of this strand, that is complementary strand 3-CTTAAG5-. These are complementary strands and these are palindromes to each other. So CRISPR works on these types of strands. Cas9 is CRISPR associated protein 9. It is an endonuclease enzyme that causes double-stranded DNA breaks. An endonuclease is an enzyme that breaks the phosphodiester bond in a polynucleotide chain, within a polynucleotide chain. Nevertheless, CRISPR-Cas9 is a gene editing technology that makes it possible to correct the errors in a genome or to turn on or off the genes. Here genome refers to the genetic material in the body, including DNA and all the genes in the body. Based on these processes, based on the ability of editing the genes, CRISPR-Cas9 can have laboratory applications as well as clinical applications. The laboratory applications of CRISPR-Cas9 may include live imaging related to the genome and rapid production of cellular models. On the other hand, the clinical applications of CRISPR-Cas9 may include gene therapy and treatment of infectious diseases such as those caused by HIV. So this is little introduction to CRISPR-Cas9 for beginners. You can also listen to the summary of this presentation in German language. Before going ahead, I would like to say that you can support me on Patreon so that I would keep on posting these types of illustrations and presentations for your information. Thank you. Ich bin Usman Zafar Pracha. Dies ist eine Zusammenfassung in deutscher Sprache. CRISPR-Cas9 ist eine Gene-Editing-Technologie. Es wird verwendet, Fehler im Genom zu korrigieren. Klinisch ist es vorteilhaft bei der Gentherapie und Behandlung von Infektionskrankheiten. Danke.